So today we're going to start to consider how proteins are synthesized within cells. We've talked previously about the fact that DNA acts as the genetic code for protein synthesis, and we know that it's the sequence of the DNA bases which acts as the genetic code. DNA acts as a triplet code, so it's the sequence of every 3 bases that's going to determine the specific amino acid that is placed into polypeptide chain and therefore producing a specific protein. We know that protein synthesis takes place at ribosomes, and what I want to do initially is look at an overview of the first step in protein synthesis, which is called transcription. So to start off with, let's have a look at a eukaryotic cell. So we know that eukaryotic cells have got their DNA wrapped around histones. So let's have a look at one of those as an example. So here I've got a eukaryotic cell. Here's the nucleus. Here are 4 chromosomes, and we've got rough endoplasmic reticulum within the cytoplasm with ribosomes on the surface. Let's start to have a look at this example. So if we zoomed in and had a look at a chromosome as an example, and we're going to ignore the histones and just look at the DNA molecule itself. So this is just a section of DNA molecule. We know that it is made up from 2 polynucleotide strands which are joined together via hydrogen bonds at the bases. So here's the DNA molecule itself. So the DNA molecule, and we're interested in the sequence of the DNA bases specifically because this is what acts as the genetic code. The sequence of DNA bases can be split into 2 types. You get certain sequences of DNA bases which are referred to as being coding. So the sequence of bases acts as the triplet code. You also get sequences of DNA bases that are non-coding. They do not act as the genetic code. We need to talk about what a gene is. So a gene is a specific sequence of DNA bases that's going to code for a polypeptide being formed at ribosome. So for example, on this particular chromosome, here's a sequence of DNA from this point just here, let's say, to this point just here. That sequence of DNA bases is going to be the gene that codes for, and I'm just going to give an example, codes for amylase enzyme. We then might have a region of non-coding DNA. So this sequence of bases that's present in here doesn't code for a polypeptide at all. But from here to here, this is a separate gene. So a gene is a sequence of DNA bases that's going to code for a specific polypeptide. So let's say that at this point just here's the gene that codes for insulin hormone, which you'll be familiar with. So the gene is the sequence of bases that codes for a specific polypeptide, and the actual point on the chromosome, the position of where you find that specific gene, is called the locus. So this is the locus for the amylase gene. A bit further along, this is the locus for the insulin gene. So we know the position of where these different genes are. So, as we said before, here is a region of non-coding DNA. If we look within a gene itself as well, so let's have a look at the amylase gene, for example. So now I'm just going to draw the DNA molecule as just two straight lines. So let's have a look at that gene in a bit more detail. But I'm just drawing it as two lines. I'm not drawing the DNA molecule as a helix. Within a gene, so here's the gene that codes for amylase enzyme. Within a gene, you also have regions of non-coding DNA. You also have regions of coding DNA. So the regions of coding DNA are called the regions of non-coding DNA are called introns. So an intron is non-coding. So introns are non-coding. Make that clear again. So that's going to call that I. You've then got regions within a gene that are called exons, and an exon is a region of coding DNA. And you have introns and exons within a gene. And this is within eukaryotic cells. We'll talk about prokaryotic cells in a moment. So that's the overall sort of structure of looking at a DNA molecule. The fact that genes are sequences of DNA bases that code for a specific polypeptide. We can call the locus is the position at which you find that gene. Within a gene, you have sequences of non-coding DNA, introns, and you also have sequences of coding DNA called exons. You also have, in the spaces between genes, regions of non-coding DNA. In terms of what's going to take place in protein synthesis, the genetic code for a specific gene, therefore coding for a specific polypeptide, that genetic code has to be present at a ribosome to tell the ribosome the sequence of amino acids to be placed in that polypeptide chain. So the genetic code physically must be present there. DNA cannot leave the nucleus. It's physically too large to exit through the nuclear pores and make its way out and be present at the ribosome. And this is why the first step of protein synthesis is to convert the genetic code in the form of DNA into a smaller molecule, a smaller nucleic acid. Remember, all nucleic acids carry the genetic code for protein synthesis. And so the first step is transcription. So transcription is going to take the genetic code in the form of DNA and convert it into RNA, and specifically messenger RNA. We know that one of the differences between DNA and RNA is RNA is smaller. An RNA molecule is just a single polynucleotide strand, and so that can move out of the nuclear pores and make its way to the ribosome. So what we've got with eukaryotic cells is our genes, which are made up from introns and exons. What's going to happen then in transcription, and this is just an overview before we look at transcription in more detail. What's going to happen then in transcription is the DNA, so this is DNA, is going to be converted into RNA, and specifically messenger RNA. And in transcription, the whole of the sequence of the DNA bases is copied. So all of the introns and exons are copied. So coding and non-coding sequences. And we refer to this RNA or messenger RNA that's been formed in transcription in eukaryotic cells. This is what's referred to as initially pre-mRNA. Pre-mRNA. It's called pre-mRNA because at the moment it's not quite ready to be used at the ribosomes in the second stage of protein synthesis, which is called translation, and it's called pre-mRNA because it contains, at the moment, those non-coding introns. In a process called splicing, what happens next, as you can probably guess, is that all of the non-coding introns are removed, and that then leaves you just with the coding sequences of DNA bases, which are the exons, in which case now this is referred to as just messenger RNA, and then the messenger RNA can be used in translation, and in translation ultimately then you will get amino acids being placed in a specific sequence. So there's an amino acid, and from that one gene, so here's the amylase gene at its specific locus in the DNA molecule, transcription will produce pre-mRNA, splicing will take place, the non-coding introns are removed, we then have mRNA, which moves forward at translation. We'll look at the details of that later on. We then get our specific sequence of amino acids, which is going to code for a specific polypeptide. So that polypeptide will form the amylase enzyme. In a prokaryotic cell, so if we just consider a prokaryotic cell, we know that in a prokaryotic cell the DNA just exists as a loop, no histones associated with the DNA itself. The process of protein synthesis is identical in prokaryotic cells, except for one feature. Prokaryotic cells are just made up from, or prokaryotic cells, DNA rather, prokaryotic cells, DNA is just made up from coding DNA. So there are no non-coding regions in between genes. You just have gene next to gene next to gene next to gene, and within a gene there are no introns. Within genes in prokaryotic cells, you just have, it's all exon, it's all coding. So that's a brief overview of the general process. What we're going to do is look at transcription in eukaryotic cells in more detail.